The broadcast is now starting and all participants are in passive listening mode. This workshop will be recorded and the recording might be shared with others. The participants list is closed and the chat function operates in private mode with only the speakers and the moderator accessing the chat messages. If you prefer not to proceed under these conditions, please log out at your convenience. Hello and welcome, ladies and gentlemen, to our mindsON RBQM workshop series presented by Syntegrity's myRBQM Academy. I'm Tamara, the Academy's Operation Manager. Today, we present a mindsON special edition, the cost optimization power of RBQM backed by data. This interactive workshop will last approximately 60 minutes and includes polls and a round table for your questions and our answers at the end. I would now like to introduce our speakers to you. Anastasia Shapovalova, with over 20 years of experience in pharmaceutical operations and business development for major pharma companies, Anna is leading Syntegrity's centralized monitoring and analytics department. Amongst others, she is renowned for her expertise in business analytics, strategic planning, risk management, SDV reduction, and operational efficiency. Sven Goracik, with broad business technology and data analysis expertise, Sven managed Syntegrity's myRBQM system for clinical trials in our project management team. He developed Syntegrity's interactive data visualizer. His pivotal contributions consistently reinforce RBQM risk management and operational efficiency. Julian Nunes-Gonsalves is a scientist by training with experience in preclinical and clinical research, marketing, and business development. He leveraged this combined expertise to overcome the common misalignments between sales and marketing teams and effectively communicate scientific content for impactful results. As Syntegrity's RBQM solutions architect, he adeptly translates client needs into actionable RBQM solutions tailored to each organization's needs. So, for those who are new to our mindsON RBQM webinar series, it is designed to be interactive. During this workshop, I will activate polls with various answer options you can choose from, and the chat will also be available. You find the chat window on the right side. Send us your questions or comments there and select the option Send to Moderators. So the speakers and I receive your questions, and later on we are going to address your questions then in our roundtable session. Ladies and gentlemen, thanks for your attention. We are starting now with our mindsON RBQM Special Edition workshop. And Julian, over to you, please.
Thank you, Tamara. Hello, everyone. Happy to be here and to be presenting this topic to you all. I hope that it's going to be of interest and at least trigger some thoughts, some questions, and happy to hear some follow-ups after this as well on the topic. So really what we want to be showing you today is take you through a number of areas that we propose through our experience, our research, that will result in cost savings when an RBQM strategy is in place. We want to show you the measurable or quantitative value of RBQM, ultimately driving efficiency through the following areas. So we're going to cover a couple things today. The most obvious one, of course, is going to be cost optimization. So things like how that's going to happen. We're going to discuss that in quite some detail today. What I'll be taking you through first is going to be the compliance advantage, as well as data quality and integration. And then some safety enhancements and time efficiency. I apologize if there's some background noise there. Unfortunately, there's not much I can do about it. So compliance advantage, we'll be talking about just why compliance or staying ahead of your compliance and being compliant. Compliance is always it's a massive buzzword, of course, and it's a contentious one. But we're going to show you why that can be a good opportunity for cost savings potentially. Similarly with data quality and integrity, safety enhancement, and time efficiency as well. I'm not going to go through the finer text here. We're going to cover all of those in subsequent slides. So the first of these that we want to talk about is going to be compliance advantage. So here what we're looking at is things like the protocol compliance, of course, so being compliant with the protocol that's been set out, and then also regulatory compliance. So firstly, with regards to protocol compliance, here we're talking mostly about reducing the number of major and critical protocol violations. We've been able to show, a 50% decrease in protocol deviations simply due to early deviation detection. Being able to detect those deviations in early on allows you to address them quicker, more efficiently, and then also implement mitigation actions that are going to be effective. You've got a system in place. Remember, everything that we're discussing today assumes an RBQM strategy is in place. RBQM does not just refer to just a technology, but or a software platform, although that's a major part of it, of course. We're also talking about processes, SOPs, etc. And one of those examples is reducing those protocol deviations. By having an early detection system in place, you can address those quicker and ultimately reduce the number of protocol deviations through continuous learning by up to 50%. Similarly for screening failures as well. So screening failures here, we're talking about a possibility of 30% decrease. And again, this is due to that continuous learning, addressing or evaluating which of those in and exclusion criteria result in the most screening failures in past studies, avoiding those or amending them for future, and thus that continuous learning process allows you to decrease the percentage of screening failures. And also, remember, this is an ideal RBQM system will have near to real-time monitoring as well. So we're going to refer to this quite a lot today. If you have a system in place where you can monitor those thresholds, and as soon as it gets to a concerning level, you address it immediately, rather than at regular intervals, which could be a series of months, it could be much longer, or it could be only at the end of the study that you evaluate, okay, what was the percentage of screening failures? By then, it could be an astronomical percentage. So this way, it's a near to real-time monitoring. You can address those and address the findings in a timely manner. Then moving on to regulatory compliance as well. So here we're talking about decreasing the number of findings, simply decreasing the audit and inspection findings, again, by up to 50%. And again, this is because you are performing this near real-time monitoring. You are able to see what could be a finding down the road and address it rather now. So those findings, or those potential findings, those data metrics or data findings that could be a potential issue, you can address it in time. And when it comes to the inspection or audit, you can already show them that this is what was done, we found this, we did this, and everything is in an audit trail accessible through the RBQM system. And then lastly on this slide, minimizing the missing primary and secondary endpoints by up to 30%. Again, like I've said, we're going to talk a lot about this, but that near real-time monitoring, that over time you're monitoring what is the percentage of missing endpoints, and you can detect those earlier and obviously address them with the site sooner, thus reducing future cases. So a lot of this is all going to be about continuous learning, continuous adaptation, adjusting your strategy and not the protocol, but the overall management of the study, the mitigations in place, etc., and activating them at the right time. It's about just having a clear oversight of your continuous study process so that you can address each of these findings as and when they appear. Then data quality and integrity. So here we're talking about simply enhancing the data quality. You are able to review the data in a timely manner. You can narrow down those confidence intervals. You can reduce the noise component because you've got access to a much better oversight, or better way to perform oversight of that clinical data. And then on the second point, reducing database reopening or thaws compared to historical data. we've been able to show, at least in our research, by up to 50%. You can decrease the number of times that you need to thaw the database, simply because, again, like we said previously, early detection of those data issues. You can identify what could be an issue way before the database is closed, so that's reducing, speeds up the availability of the study results by up to several weeks in some cases. And then safety enhancement. This is quite intuitive, I would think. Here we're talking about enhancing the subject's safety measures, simply decrease in the delay in SAE reporting due to, like we said, the tracking the adherence of the SAE reporting timelines. Remember that you have a system in place with key risk indicators, notifications that are going out to the users to say that this particular site, this particular region, whatever it might be, they might have a poor record of adhering to the AE or SAE reporting timelines. So you can address those much more quickly and a lot more stringently as well, because you've got the data to support your findings. So again, you can decrease from previously an average of 48 hours to 32 hours or even less, depending on what your processes dictate, of course, but you can just improve the adherence of SAE reporting timelines. And then also the response to SAE related queries. These can be 75% faster. Like I said, this is because you've got a near to real time monitoring platform in place where you've got alerts going out, and SAE is noticed, it's submitted, reported. The teams can immediately react to that. So a much, 75% faster response is quite considerable. Then here we're talking about the improved consistency as well. Consistency is a major concern. If there are inconsistent reports, inconsistent findings, or even the way in which they reported, simple nomenclature, wording can be changed. So at least here you have a process where you can improve the consistency, identify any under and over reporting, and ultimately result in a more consistent safety profile for the new entity. And something I've been mentioning a lot, but the near real-time monitoring, in this case, medical monitoring, avoiding those late safety-related surprises. And of course, this should be very, very important. It should be a priority for any clinical trial. Patient safety is a very big deal. It should be, at least in my opinion, the primary concern at any time. We want to make sure that the patients and the participants are always our top priority. So avoiding those late safety related surprises where it could already be considerable detriment to the participants. And then time efficiency, also something that's fairly straightforward, accelerating the study conduct timelines. By simply having this process in place, you can accelerate the study conduct timelines because you have the ability to preempt any concerns, address them, mitigate any risks. Remember, we're talking about RBQM here, which is risk-based quality management. So everything comes down to risk proportionality, applying the most effort where the most risk is. So here, any major risks to the study conduct timelines can be addressed much more effectively, much more quickly, and ultimately just accelerate those study conduct timelines because you're not going to hit any roadblocks, or at least you will know what roadblocks you're going to encounter. Reducing the time from last subject, last visit to database lock. This is, again, ongoing oversight simply results in no loose ends, and database lock can be done within 2 weeks or less. Bear in mind, this is not considering any other interim analyses or lab data that you're waiting for. This is a best case scenario, of course, but there may be other parameters that may increase this, not related to the RBQM strategy. And then enhanced predictability of study enrollment and resource planning for database closure. What we're talking about here is just being able to predict, making sure there's no over enrollment. What is my enrollment rate going to look like? How many patients do I need at key milestones of the study timeline? Improve planning of those interim analyses, making sure that the interim analysis databases are available within the 2 weeks needed. And it's just giving you oversight over the whole study. What is my enrollment timeline going to look like? And allocating your resources effectively. And then, of course, we get to probably the most interesting part and much awaited by everyone, I'm sure, the cost optimization. So cost optimization, we're going to look at a couple areas here. So risk management is going to be the first one, operational efficiency, reduce source data verification. How does optimizing the visit schedule affect this? How does reducing the protocol amendments affect cost optimization? And then lastly, the increased compliance as well. So with that, before I hand over to Sven, who will take you through this section, we have our first poll. So Tamara, if you can pull up the poll, I'll just read the question first. So the question we want to ask here is, of those that we just discussed, which costs can be optimized the most by implementing RBQM? Is it going to be non-compliance? Sorry, just going to wait for the responses to come back. So non-compliance induced costs, protocol amendment induced costs, SDV and monitoring induced costs, data quality, or operational inefficiency. If you can just give me, we'll give you a few seconds to put that in. It will be interesting to hear what you think would be the biggest cost saver.
Okay, just another moment before we close the poll. All right, I'm presenting the results now. Thank you very much.
Great. All right. So we've got, of the 28 people that voted here, vast, I wouldn't say vast majority, but almost 50%, 42% said C, SDV and monitoring induced costs. Then next was 28% D, data quality improvement costs. And then an equal 10.71% was E, operational efficiency, and then B, protocol amendment. And no one voted non-compliance induced costs over here, which is interesting. So Sven, over to you to take us through what the actual values say. And I think everyone will be happy to hear that they're on the right track, I think.
Thank you, Julian. So as Julian showed in the previous slide, the starting point toward clinical trial cost optimization is doing the risk management in the first place. So this slide here, the graph on the slide shows a clear positive correlation between the amount of rework in a clinical study with the cost incurred as a result of that poor quality. Or in more simple terms, logically, the more rework needed in a study, the more expensive the study gets. So along the curve, we have outlined a couple of examples of such negative study occurrences from minor ones, such as unexpected shift from one to other hemisphere in search for appropriate patients for, say, seasonal indications, to more critical ones, such as major sample size increase, major study delays, and in worst case, study repetition. It is also important to note the exponential nature of the curve, which reflects the tendency of this risk and issues, if left unattended, to quickly snowball and escalate into much bigger issues that need later on much bigger resources to be rectified. Which basically brings me to the value of doing the risk management in the first place. So by implementing RBQM and really proactively thinking about preventing risk, for example, by utilizing RACT, Risk Assessment Categorization Tool, you are able to identify critical data and critical processes and related risks to those critical processes and data, and in turn, design mitigation strategies way before first running trial issues emerge. After that, you need some kind of mechanisms and tool in order to detect those risks. Namely, some risks are, of course, easy to detect, especially for experienced investigators, such as patient enrollment not going according to the enrollment plan. However, some risks are more hidden, much more hidden, and really require data and data analysis to compare trends to catch these outliers and anomalies. So the tools such as key risk indicators and quality tolerance limits can help you do exactly that. Detect, evaluate, and control these risks. So after you were able to do what I previously described, you still need a process and tools in place for effectively and efficiently communicating and escalating those risks to the appropriate staff within a study. So majority of modern RBQM softwares out there have this kind of issue management or ticket module that allows you to do exactly that. Automatic ticket and query generations that will be assigned to appropriate staff in a timely manner, ultimately allowing you to have a full overview of all items and issues within the clinical study that are still left unaddressed. So moving to the next slide. Let's pick up my pointer. So as a result of implementation of all these previously described risk management practices, we were able to outline three most significant sources of study conduct savings. So as Julian discussed in the poll question, first and most significant ones is site monitoring costs, which we evaluate by our conservative estimate that we can shave off 10% of those by implementing RBQM, resulting in $1.5 million in savings. These cost reductions are mainly derived from reduction in source data verification and ability to optimize monitoring schedule by targeting those sites that communicate or report more deviations and escalations, and perhaps skipping some monitoring visits for the sites that seem to have everything under control. Furthermore, early detection via RBQM results in reduced protocol amendments due to overly complex protocol and early participant dropouts from an average of 3.3 protocol amendments per study to 2.8 protocol amendments per study, resulting in a save of $250,000 US. And lastly, avoiding to repeat the study due to vague outcomes will save you anywhere between $200,000 and $800,000 or more. So, of course, not two studies are exactly the same, and the extent of the savings from each of these sources cannot be implemented to each study. So, however, by averaging these three savings, we believe that we come to very conservative and very reasonable estimate of saving of at least $750,000 per study. And now before introducing the next source of clinical cost optimization, I would like to ask you, in your opinion, at minimum, how much sooner can you expect your product to reach the market by implementing RBQM?
And the poll is open now. You can select one answer, please. So thank you all very much. I'm presenting the results now. And here we are.
So I can see that majority of people answered C, which is nice to see that there are so many RBQM believers and supporters on this webinar. However, the answer that would be in alignment the most with our conservative estimate, and there is a hint in the question itself with the at minimum part, would be a 1.5 weeks. However, now in the next slide, I will hopefully present why this is not negligible at all.
Sven, excuse me, please, before we come to the next slide, I have a question here that we can maybe address now. Could you please repeat the source or context for your analysis? How many studies were included?
Yes, the source context for our analysis are our use cases. So the studies that are run with RBQM and without RBQM, and exact amount of studies can later on hopefully be answered by Anastasia, who helped with the actual analysis.
Thank you very much, Sven.
Thank you, Tamara. So another source of risk management fueled source of clinical trial cost optimizations are market access savings, which refer to the excess of revenue that you're able to reap because of the exclusivity benefits of your products due to its patented life duration. So before moving to the figures below, it is important to note that for these figures, we assume a 520 million annual sales product. And for reference, AbbVie's bestseller, Huspira, I think it's called, that treats adolescent idiopathic um, arthritis, uh, yielded annually between 2014 and 2022 between 8 and 21 billion annual sales. So based on our experience, based on our analyzing use cases, but also previous experience from some of our senior medical and scientific officers, we were able to outline 4 of these most common causes of delays in going to the market. And with that, we were able to kind of appoint an average duration of the delay for each cause listed here. So for example, major study delay due to update on participants lost to follow-up. On average, delay is going to the market by 4 weeks. And by implementing RBQM and all of the best practices, which some of them I discussed earlier, you will hopefully be able to create mitigation strategies for this participants lost to follow-up issue and ideally tackle all 4 weeks of this delay, saving you ideally $40 million. Similarly, with implementing RBQM, if you are able to avoid major sample size increase due to many in- and exclusion criteria violations, poor adherence to treatment plan, or poor compliance, that could save you 1.5 weeks of delay, saving you $15 million. Likewise, on the point number 4, avoiding unexpected shift from one hemisphere to another due to slow enrollment in the case of seasonal indications can avoid 4-week study completion delay, which would result in $40 million saving. And lastly, rework to improve data quality due to too many missing endpoints or data outliers towards the end of the study. From our experience, on average, cause additional delay of 2 weeks between last patient last visit data to clean database. We are, of course, aware that there are many more variables that kind of go into this delay between last patient last visit data to clean database, but the one about reworking to improve data quality we can tackle with RBQM. So like it was the case with the study conduct savings, of course, not all of these market access savings can be implemented to each study. So if you are able to implement even the smallest one, you will be able to save at least $15 million per product with assuming a 520 million annual sales product. And now after risk management, to introduce second source of clinical trial cost optimization that derives from operational efficiency and better compliance. So this basically assumes all of these functionalities that modern RBQM softwares have that traditional tools such as Microsoft Excel, for example, do not have. So functionalities like daily real-time data transfer that allow you early detection of risks and issues, full audit trail that Julian mentioned that allows you better traceability and therefore compliance, uh, functionalities such as change monitoring that allow you optimizing the monitoring costs, built-in RBQM workflow are all the sources of this operational advantage. So we outlined costs associated with FDA inspection findings, or more precisely Form 483, more commonly known as FDA warning letter, as an example where functionality such as having a full audit trail really comes in handy. So resources indicate that up to $400,000 can be wasted in responding to FDA warning letters, while non-compliance can cost you anywhere ranging from $250,000 and up to $5 million in a worst-case scenario of study cancellation. So conclusively, our estimate that a bad FDA warning letter response may result in a cost for your company of approximately $300,000 per study. And now regarding cost optimization from visit schedule, you will hear more from Anastasia.
Thank you, Sven. Good morning, good afternoon, ladies and gentlemen. I'm going to discuss with you today two opportunities, and the first one is two opportunities to reduce your clinical trial budget if you implement RBQM. And the first one, where what I'm going to talk about is reduction or optimization of CRA regular monitoring visit. Where's my pointer here? Regular monitoring visit, what you can achieve in your clinical trial. So it is very well known that FDA and other regulatory services, they push sponsor a lot to reduce number of fields to be SDV'd as much as possible in order to achieve the minimum price in the future registered product, if it will be registered, of course, because from field work perspective, CRA regular monitoring visit is one of the biggest part. If you look at the right side of the slide, could you, colleagues, please stop moving the cursor? Thank you. If you look at the right side of our slide, you can see the graph that represents results of the simulation, mathematical simulation, what we made internally, and what showed that for different study sizes, even if you implement very conservative assumptions for risk-based scenario, you will be able to achieve up to 10% savings versus non-risk-based scenario, which in absolute values means like $100,000 in small size studies like oncology, and several $1 million if we're talking about big studies like diabetic and cardiovascular. And on the next slide, I will go through this calculation and I will explain assumptions what we made on an example of oncology study. So let's go to the next slide. So I should mention a couple of things. First of all, we published this calculator on our website. It's available for free, so you can use the link under this mask, and after this webinar, you can go and check potential savings for your upcoming studies. And also in this slide, in this part, I will cover on-site visits, but in our calculator, remote visits presented as well. I will not cover them because of time matters, but the logic is will be absolutely the same. So as I mentioned, I'm going to speak with you today on this slide about oncology study. In oncology study, we have 30, 40, 50 sites max in average. And for my particular example, I decided to use 40 sites. And if we first go to the non-risk-based scenario, we should look at our site monitoring plan, where we should find recommendations of regular monitoring visit frequency. Usually companies use from 6 to 8 weeks frequency for on-site visit. I took 7. And also I just made the quick investigation. So for the on-site visit cost, I used $3,000 US. This is it for non-risk-based scenario. And now we switch to risk-based scenario. Here we should have to perform 4 steps, and I will explain what we need to do. So first of all, we need to split our sites into 3 baskets. So low risk sites, medium risk sites, and high-risk sites. Ideally, you have history, some previous experience with these sites, how they performed in other previous studies, what kind of issues they faced, how often these issues occurred, how fast they solved the issues, if these issues occurred again, etc. This is an ideal situation. In best case scenario, you have minimum 10 studies for each site in your past. So using this information from previous studies, you can understand what site created a lot of issues, what site created average number, medium number of issues, and what site were not problematic at all. If you do not have historical information, you usually refer to the subject matter expert who can provide you some feedback on sites in your what you are planning to what you are going to include into the upcoming study. And in the worst case scenario, you do not have any information about this site. And in this case, you have the only option, which is using of open sources. In our case, such open sources can be clinicaltrial.gov. I would recommend you to check previous 3, max 5 years and check if the site, what you're going to include into the upcoming clinical trial study participated within the past 3-5 years in any similar study. Similar, I mean similar therapeutical area or similar condition. So that's not a kind of assessment of a site. It's a kind you should treat it as a good or bad signal about site potential. Of course, it's not as good as in case of historical analysis of site performance, but if you have no information, what can you do? This is the only option you have. In real life, most probably you will have to combine all of these 3 options. Then we, on the next second step, we need to understand minimum amount of work, what should be done, and minimum site visit frequency. What we then can apply to the low-risk site, because from low-risk site we do not expect a lot of issues, or even if issues occur, they are not major and they can be easily managed, etc., etc. So how we define this frequency? We know. On the first step, we already defined how many sites we're going to have in a low-risk group. We know how many patients we expect from these sites. We also know what enrollment speed should be. And we know what kind of fields are needed from SDV forms, from CRF's forms, sorry, in order to analyze outcomes and other crucial information for the product registration. So that's all of these things allow you to understand the amount of work which should be done in each study day for average low-risk site. And also, probably average capacity of your CRA. If you don't know average capacity of your CRA, we can recommend you to make a test, minimum with 3 people. Just lock them for 8 working hours in a room and ask them to perform regular CRA work, in order to understand how many pages for a particular study CRF forms they can manage during the working day. Maybe some other procedures also should be done. So double check if something is dramatically wrong in your process. You will see it immediately. If not, at the end of this testing day, you will understand, based on this minimum 3 people examples, what is the average capacity for a particular study. I will not give you any specific references here because I interviewed several companies from small size companies to big pharma companies, and they all gave me different references. And we should understand, of course, that first of all, it depends on your protocol complexity, your CRF complexity, your SOPs complexity, and some other guidelines. So this is very, very specific thing. Anyway, we got all needed information, perform all needed tests, and we calculated minimum amount of work. We understand average capacity of our CRA, so we can understand with what frequency we need to visit low risk site in order to cover minimum amount of work. This is our second step. Then we should go to the medium. Then we should decide what frequency should be for the medium risk and high risk sites. And for this purpose, we should understand, if we go to, if we use medium risk site as a reference, what kind of problems we should expect, we can expect from medium risk sites. Here again, of course, you need to check your protocol. You need to look at your CRF and the other documentations. But what kind of problems we expect in oncology studies? Of course, it's adverse events, serious adverse events. It's images, labs, lab tests, CONMETs, and in all kind of studies, no matter what therapeutic area, it's always data management, including query management. So look at the sites what you have included into the medium risk group at step number 1. Try to understand how often they can face such problems based on their experience, based on your knowledge from previous studies, etc. And compare amount of additional work, calculate in working hours additional amount of work what a CRA will have to perform at this group of sites, and add this amount of work to the minimum amount of work what we defined at the step 2, and then calculate percent what will show you comparison between low risk site and medium risk site from time perspective. And we should make the same exercise for the high risk sites. When we define this percentage and put them into the corresponding fields for medium risk sites and high risk sites, calculator will propose you with needed frequency in order to cover this additional work what CRA will have to perform in different risk in different sites from different risk groups. This is our third step, and now we go to the step number 4, where we have to speak about money. So of course, no matter what kind of site we're going to visit, we always have basic costs, which are travel, food, some other things. Double check your compensation policy and understand how much money you will have to include anyway into the visit cost. Then we go again to the low risk side. We know from the step number 2 what amount of work should be performed in order to cover minimum amount of work to be done at the low risk side. We know how much days or hours, if you are lucky, a CRA will have to spend on this site, so you can calculate what will be the cost of this time for the CRA. Then you combine all these two things together, and you have your cost for the low risk side. Maybe your finance department will tell you to add something else. Double check your policies, your guidelines. When we go to the medium and high risk side, there for medium risk side, we already calculated at step number 3 additional amount of work, and we know how much it will take in percentage. So we can take base cost, which is travel, food, etc., minimum amount of work increased by the percentage what we calculated at step number 3 for the medium risk site, and we recommend you to consider minimum 1 training session for medium risk site, because these sites, they have problems and you will have to manage them somehow with the site team. You will have to read them some additional trainings. You have to remind them about SOAPs, how to adhere the SOAPs, etc., etc. So you need to add this time to the visit cost. And for the high risk side, you do the same math, but we recommend you to consider minimum 2 training sessions, because high risk side, they have a lot of problems. You will have to face them permanently. You will have to manage them mostly on each visit for this site. So that's how we calculate visit cost in each risk group. And when you finish with the step number 4, the calculator will provide you with potential savings in case of implementation risk-based quality management into the CRA monitoring, regular monitoring list. In our case here, we see that savings are around little bit more than $200,000, which is a little bit less than 10%, which is in line with our previous with our simulation, what I showed you in the previous slide. This is it for the CRA work optimization, and now we're going to the next thing, which is the reduction of protocol amendments. So there is one regular research which is performed by Tufts University Medical School University team. I saw that they make publications almost every year. And in this publication, if you check the latest available publication, you can see that they found very warning tendency that protocols, what experience, a percent of protocols, what experience minimum 1 protocol amendments is growing for the last decade from 69% to 76%, and which is more important, average number of protocol amendments per study is also growing from 2.4 to 3.3. We should remember that protocol amendment, it's not an easy thing, and it's also not a cheap thing. So 1 protocol amendment will cost you from 250 to 550K US dollars, which means that if you are super lucky, you can add $1.5 million to your study budget. Finance director will not be happy about this news, and it's a problem. So, but there is another finding from Tufts University team. They found that up to 30% of protocol amendments are avoidable or partially avoidable, which means that if we have a system what allows us to monitor, to understand in what kind of in what percent of cases protocol amendments happen because of regular errors in protocol, we can limit this number of changes in the future studies. How can we do it? We can have a system where we upload our studies, as many studies as we can, and we have a list of risk indicators which exist not because they're risk indicators, but because they help us to control risks in these studies. It's very important. And also, in addition to these risk indicators, what allow us to monitor study situation on daily basis, if we can do it from technical perspective. We also should have a system what allow us to manage issues. So what happened with in each study, if risk indicator send us some alarm, and we cannot. This system should not only notify us, the system should allow us to create the investigative corrective actions and monitor their effectiveness. And then if we understand that for some bunch of issues, protocol design was the root cause, we can collect this information, we can collect potential problems with protocol from bunch of studies, and then we can share this information with the team what designed the protocol for the future studies. The problem is with this exercise, it sounds very clear what to do, but the problem with this exercise is that it's not something what you can do over one night. You will have to spend from several months to several years in order to collect needed amount of information. It depends on your protocol protocol complexity. It depends on enrollment speed in each studies. It depends on how good you are in issue management and root cause findings. So it depends on many things, but it's doable. And that's how a risk-based quality management system can help you with protocol amendments reduction. So that is it from my side. I hand over to Julian.
There we go. Sorry about that. It took a while to come unmute for some reason. Thank you, Anna. That was very, very good. And thank you, Sven, as well. So with that, let's summarize before we go to some questions and answers then. So really, in this graphic, we can take that we looked at earlier, we can look at exactly what this comes to. So risk management, we can look at about 750,000 in savings. Combined operational efficiency and increased compliance can be about 300,000. And as Anna was demonstrating now, your reduced SDV and optimizing the visit schedule is about 1.5 million. And then lastly, reduced protocol amendments, about 250,000. So all in all, this comes to, by our estimates, 2.8 million per study, which is considerable, of course. Then also don't forget the market savings as well. That's 15 million per product. A comment came in earlier, of course, that's only if the product does go to market. That is, of course, true. But again, I mean, the savings there for a product that is going to go to market, that is considerable. Bear in mind that this 2.8 that we're talking about here, it doesn't take into account the effects of cost savings related to all the other items we discussed earlier as well. So things like decreased protocol deviations, screen failures, database thaws, etc. So all of those can result in additional savings as well. So keep that in mind throughout. And of course, this always comes with some kind of caveat, based on, and I'm sure there's some questions about that, which we'll respond to in a second. How did we come to this, which we'll discuss in a second. But this is based on really our findings, our research, and we'll discuss a little bit more about this in the Q&A as well, because I know there's some questions about that. So let's leave that for then. I also wanted to just say this will also vary, the savings here will also vary depending on operational practices, therapeutic area, etc. As I said earlier, what we're saying here is that everything is it's an RBQM strategy, not just an RBQM tool, but making sure that you have an RBQM strategy in place, the right processes, the right SOPs, etc., the right training as well, and that's a crucial element. So people, technology, processes, these are all factors that come into play. So with that, I think let's go to some Q&A, and I'm going to hand over to, Sven and Anna. If there's a question that you'd like to address first, please feel free. So let's start that as we wind down the webinar.
Well, Tamara, please correct me if I'm wrong, but in my chat, I cannot see any more questions.
No, as far as I can see, we've addressed the ones that we got during the webinar. So, dear participants, if there is anything you would like to ask, then feel free to put it in the chat now, and we can talk about that, please. Or just give us your feedback, what your opinion is. So we are having a round table now.
Did we might have maybe done this privately to the person who asked, but I think it's a good question to just quickly give a response to. A question was asked, what's the source of this data? Is this published and available in a publication? It's not published. There is a number of studies that we've used. There's no specific number of studies that we've used. This is empirically derived, right, from seeing the risks that in our system, the risks being addressed early on in the process. We've seen the benefits hands-on. So an issue also is always that, customers understandably are hesitant to provide the data based on prior to using the RBQM system, so it's hard to do a comparison. But empirically, this is very clearly a cost savings benefit. And this could be a follow-up discussion as well. There's been discussions internally about maybe a round 2 for this as well. We might have a panel or something as a future webinar on this topic. But yeah, that's just something I wanted to add in there based on a question we had. I don't see any other questions coming in. Sven, Anna, anything you want to maybe add to close us off?
I can add from.
Okay, we lost Sven. There we go.
Yes. Okay. So the question is, or comment, that benefits appear to be for large-scale trials, and is there similar impact for smaller BMCFs or device trials? So, of course, these benefits grew larger as the number of studies and the size of the study gets bigger, but especially regarding the SDV and site monitoring costs. However, the benefits that were discussed from Julian regarding compliance, regarding decreasing the number of protocol deviations are appropriate even in the smaller studies.
I would say that here study size is not so as important as your experience with using RBQM, and that you should set right expectations to each group of sites. So especially for medium and high risk sites, you should properly define what site should be in what group. So in absolute numbers, savings will be for small studies. In absolute numbers of savings will be smaller, much smaller than in case of big sites, but in relative assessment, like per percent, it's as we showed, it's 10%, even with very conservative assumptions. So it requires experience, it requires testing, it requires follow-up and understanding what we did wrong and how we can do it better next. So that's the key issue from my perspective, the key. Answered. Okay, for question to answer.
Yep.
Thank you, Anna. And I see that majority of questions still coming in are of the similar nature regarding the difference, how much these RBQM driven savings can be achieved for bigger studies versus smaller studies. So I think that we address those.
Yes, and it looks like that was it. No more questions we have not addressed. So I would like to thank you all very much for attending this session here. And before you leave, I would like to invite you to our next mindsON RBQM workshop. That's the episode number 24, presented by our well-known speakers, Johan Pröve and Keith Sorakut. And that's just around the corner. So this Thursday on the 25th of April, will AI really unlock the power of RBQM? This Thursday, and it would be our pleasure if you join us again then. Sven, Julian, Anna, thank you very much for this great insight. Ladies and gentlemen, if you have any question, or you would like to send us your feedback or anything, you can always contact us at academy@syntegrity.com. It would be our pleasure to hear from you, and see you again soon then in the next webinar.